my presentation is too technical or, trying to get some feedback. Anyway, so that's what I wanted to ask for. You wanted me to talk about working as a biologist in the industry. That's funny because I'm neither a biologist nor am I working in the industry. So that's the funny thing, right? So I actually am trained in physics. I'm doing biology research, but I'm doing research in a university setting. So that's my disclaimers. But we'll take it from there. And still, I think my experience might be useful for those of you who are interested in working in the industry or in the private sector as a biologist, because I do work and collaborate with these kind of people a lot. And I have several biology students in my own group that I'm supervising. So hopefully their experience might be relevant for you guys. I'm going to give you a little bit of background and talk to you about the things that I'm doing at the University of Chicago in our group. And if you have questions, please go ahead and ask them. I hope that this would be as interactive as possible. So I will share my screen now and take it from there. So hopefully you can see my slides, right?
Yeah, we can see them.
Great, thank you. So as mentioned, I'm working as an academic researcher at the University of Chicago. And I started here in 2016. Before Chicago, I was at ETH Zurich in Switzerland. That was my first job as an independent lab leader as a professor. And before that, before 2011, I was at Stanford Bioengineering Department as a postdoctoral researcher. And before that, I did my PhD in optical engineering and physics in University of Arizona. I'm trained in physics from Marmara University, so this is where I'm coming from. And I have switched to doing biological research, bioengineering research in around 2008 during my postdoctoral studies at Stanford. So I'm actually not a person that is traditionally trained in biology, but as you probably are aware, there's a lot of overlap between biological research and engineering as well as physics. And I'm working at the interface, and therefore I come from a different background, but I'm already in quite a bit of in the weeds with biological research when it comes to immunity and infection and generally cell biology topics. So today I will tell you a little bit about the projects that we're doing, and I'll jump right into it and tell you about the single cell work that we're doing. So who has seen this paper before? This particular paper published in Cell by the groups of Aviv Regev and Steve McCarroll. If you're in biology, you need to know about this paper. Has anybody seen it? Can I see hands? Okay, great. I don't see any hands. So this is a very important paper because this started the revolution of single-cell RNA sequencing. And in about 2015, this paper published in Cell. So what we have here is, this is a microfluidic system. This is a drawing of a microfluidic system. These are really small channels where you have a water flow coming and being pinched by two oil flows coming from top and the bottom. And you're creating essentially an emulsion. In this T-junction, you're breaking apart the liquid flow into really small water droplets, as you can see. These droplets are about 100 micrometers wide. They are very small, and they are capable of enclosing beads or cells. And then you can do biochemistry in these. Okay, so this is the main pipeline of single-cell RNA sequencing. It's called droplet sequencing. So what you do here is you have cells coming from either sides, Then you have these beads that contain a short DNA strand that does a particular job, okay? So this encodes for the bead identity. It also captures the RNA that will be released from the cell. The cell is going to be lysed in this droplet. So when we capture a cell and the bead in a given droplet, and then have the right enzymes in the droplet, we're gonna lyse the cell. The contents of the cell will be burst into the droplet, and the mRNA molecules will be captured by a poly A tail on the DNA oligos on the beads, okay? And from that point on, we can then release the beads, amplify them with PCR, and take them to a sequencing readout. This gives this technology this unique ability of capturing individual cells, measuring their RNA content in really high throughput. You can do this in thousands and thousands of cells. And this has become a revolution essentially in biological techniques in the past 5 years, 5 to 7 years now. So why single cells? Because it has been recognized in biology, and I worked in this field before, that your typical measurements with PCR or Western blots or whatever that contains millions of cells, typically, this population average measurement is not a good measure of what's happening at the single cell level, because every cell is different. There's a lot of variability and there's a lot of noise, okay? So the field has recognized that it is very important to measure individual cells. And this technique has made that possible in fairly standardized, simple, and high throughput means. By high throughput, I mean many cells being measured in one experiment and many thousands of mRNA molecules being measured in a single experiment, okay? So this has been a very important tool, and I think anybody who's doing biological research these days should be familiar with this technique and all the computational pipelines that came after that, and all the applications of single cell sequencing in biological research. Now, so this is about measurement of mRNA molecules. And as you know, in the central dogma of biology, gene synthesis, say activation of a gene causes mRNA molecules to be made. mRNA molecules lead to protein synthesis, and proteins are the ones doing the biological actions in the cells, right? So while we have really good and high throughput means of measuring individual cells, we do not have currently a counterpart of this technique that works at the protein domain. So basically what I'm saying is in biology, we are interested in measuring proteins, but because of lack of techniques, we are using mRNA as a proxy for protein abundance in single cells. So what we are doing in my laboratory is coming up with a technique that is a counterpart of single cell sequencing that would measure proteins instead of only mRNA molecules. And this is one of the major projects in our lab, which we're pushing. Okay, so why do we need single cell proteomics? So it's been recognized, and we have also done some work on this, that mRNA molecules and proteins in a given cell are not well correlated. You will have some in cases in certain cells where you have a lot of mRNA, but you will have very few protein, and vice versa. And if you were to plot these things, they don't really lie on a nice correlated single line that goes in the diagonal. So proteins and mRNAs are often not correlated, and there are reasons for that. We can get into that later. And as you know, almost all the traditional knowledge in biology about cell types, cell functions, has been basically developed using protein signatures. When we say a T cell is a T cell because it expresses particular types of proteins, a dendritic cell expresses another set of proteins, and we basically know cell identities based on proteins. So it is very important to be able to measure these. Moreover, a lot of biological functions, for example, signaling, works with protein complexes. So you might have the expression of a given protein You might have mRNA for the same protein, but these signals tell you nothing about whether these things are in a complex, whether protein A is in complex with protein B. So it's important to measure these things in order to study signaling, and signaling is very important to understand immunity, drug development, cancer, and so on. And many drugs act directly on proteins, and it is important to measure them. So what I'm trying to tell you in the past 10 minutes is that it is very important to develop a technique that allows us to measure proteins in single cells in highly multiplex fashion in a simple and high throughput pipeline. So this is what we're doing in this project. Essentially, we call this proximity sequencing, and the paper is in bioRxiv. You can access this as we speak. So what we have is a probe panel, and the probes are made of antibodies that are functionalized with a specially designed oligo DNA strand that has a PCR handle, a unique molecular identifier, a barcode for protein identity, and a connector region. Okay, so when, for example, in a given protein or protein complex, antibody A and B bind, these DNA strands can be hybridized through ligator, a ligation connector, and then we can PCR amplify these and sequence them using Illumina sequencing or any other technique that you have in the lab, using the existing single cell pipelines. So these techniques allows us to measure, therefore, proteins, protein complexes, as well as the transcripts, the mRNAs, in many cells using a single readout. Okay, so this is a unique capability that is in the field, and because of the ability of measuring 2 proteins at a time, our multiplexing throughput scales with n squared, n being the number of probes that we have. So as an example, if I have 50 probes that I made in a cocktail and I target these cells, I can measure 50 squared over 2, roughly speaking, 1,200 protein complexes. So all of a sudden, we can measure thousands of protein complexes in individual cells, along with their transcripts. Okay, so this is a technique we've been developing for a while. Here's an application of this in a mixture of T cells and B cells. These are immune cells from mouse. So what we are doing is using some single cell sequencing analysis pipelines like t-SNE clustering to show that we can cluster cells based on mRNA as well as protein expression, and they overlap with each other. We can measure protein complexes, for example, in a given cell. This is the data that you would generate from a cocktail of 8 probes. These are the highly expressed ones, and these are the lowly expressed ones. We can do that. We can increase the number of probes that we have in our complex, in our cocktail, measure up to 30 or 40 of them. And then these would be the expressed protein complexes in each cell. This is essentially the map from one cell. And so this is a technique that allows us to measure protein complex formation, protein dissociation, expression of proteins in single cells, one cell, and in thousands of single cells under different conditions. So this is a big project in our lab. We have applied so far this to primary macrophages from humans that are under toll-like receptor signaling regulation. We applied them to PBMC cells. We have discovered new protein complexes. We have seen dissociation and association of proteins. So this gives us a new ability of looking into single cell protein expression and complex formation for the first time. So I haven't mentioned, but my laboratory does both basic science work as well as technology development. And this is one of the technologies that we are very excited about. And we are developed it, and we will be using this for studying inflammatory signaling in diseases like IBD, inflammatory bowel disease, Crohn's disease, and so on. And if you have any questions, I can take them right now, or I'm going to switch to another project and tell you about that one. So do we have any questions? Okay. Okay, so let me switch gears and talk about another project. So we are using these kind of tools that we develop in the lab and apply them to various problems, typically in collaboration with our partners at the medical school or the biology departments here at U Chicago. And one of them has been really infectious disease research. So one of the examples of this is the condition known as sepsis. So in sepsis, typically what happens is people develop an infection, and the infection ends up going into the bloodstream, creates an inflammatory systemic response. And this response, characterized by secretion of a lot of signaling molecules like cytokines, ends up becoming very deadly. Okay, so this is also called blood poisoning. This is the old school terminology. But in the case of sepsis, hundreds of thousands of people die every year in a country like the United States. And the cost to the public health system is extremely high. We're talking about tens of billions of dollars every year for the treatment of sepsis. And sepsis is very fast. So if people develop these bacterial, typically, but sometimes viral or fungal infections that go into the bloodstream, within 2 days, they can go into sepsis and then go to septic shock ultimately. And the current mortality rates are about 30 to 40%. So it's very deadly. And an interesting aspect of sepsis is the current disease progression in COVID-19 can be described as viral sepsis. Essentially, people are going into a septic phase in the later phases of COVID-19 that is due to a viral infection. So sepsis can be due to bacterial infections or other. And there are a lot of similarities between COVID-19. Of course, as we know, this has been the topic of concern for the past 2 years, but sepsis has been around for a long time, and it has continued to be very deadly. We're talking about 30%. Currently, the doctors will use some criteria that is based on rather non-quantitative sort of measures, such as body temperature, heart rate, respiratory rate, maybe white blood cell counts, and ultimately make a judgment whether somebody has sepsis or not. And that's going to determine how these people are treated. Okay. So what we try to do is really improve this and be able to come up with a much more accurate diagnostic tool based on measurement of certain molecules in the blood, so that people can be diagnosed accurately, diagnosed quickly, and can be treated differently in the case of sepsis. So what we did in this particular work, even before we started working on sepsis, is we used the previously described protein measurements. I just mentioned you this protein measurement technique we developed for single cells. We applied that to measurement of cytokines and other molecules, and we can get a limit of detection of almost 1 femtomolar, which is really, really sensitive. And we started applying this to diagnosis and monitoring of sepsis. So what we're doing is we're collecting samples, typically blood samples from patients in the ICU, into the intensive care unit of the University of Chicago Hospital. We're measuring from a very low volume of blood 2 cytokines. These are inflammatory signaling molecules, IL-6 and TNF-alpha. and we're also measuring bacterial nucleic acids, okay? We make these measurements over time, and then we feed all of this to a machine learning model. This is essentially a statistical model that learns the features of the data sets to make a prediction about patient outcomes. And since we have already followed these patients, we can develop this model using our data set. And now when we apply this to sepsis patients in the ICU, we can group these patients based on expression of various cytokines and chemokines in groups of patients who will die and patients who will survive on the right quadrant here. And in the group that will survive, we can identify who will recover early and who will recover late with very good accuracy. As you can see here, the group A is the ones that will recover early, and we have only misidentified 2 patients here out of 32, this one and this one. So basically, by making these very sensitive measurements and using some computational tricks, we can predict in the ICU who will die and who will survive from sepsis. And these patients then can be treated differently, perhaps with, depending on their progression. Antibiotics is very typical. People, doctors always apply antibiotics, but sometimes some patients might benefit from application of corticosteroids in the later phases of the disease, because it's the inflammation that is really killing them. So we can perhaps identify those patients who would really benefit from an intervention like that. So in the future, what we're trying to do is we're trying to validate this study, which was done on 32 patients in a much larger cohort. We're also developing a device that can make this measurement in much faster fashion, with much less blood, or hopefully from a finger prick, depending on how it goes. And then ultimately also determine the source of infection very quickly so that we can make an impact in this rather deadly condition. Any questions on this sepsis-related work? Okay. So we have also done some work on the study of the human microbiome. And this is in collaboration with our microbiology colleagues at the university. So as you know, we have a lot of bacteria, viruses, of course, but mainly in our guts, we have tons of bacteria that are doing certain functions, they are there, you have them from birth. And depending on how the gut microbiota changes, you might show different sort of preference for different drugs, certain foods you might tolerate or not be able to tolerate. So they do a lot of functions. And there's also a brain-gut connection. And it is important to be able to analyze these gut microbes, and these are typically anaerobic microbes, in culture so that we can understand how they change, what they respond to, and so on. The typical technological solution to analysis of human microbiome is, well, you collect some specimens, you try to grow them in culture. Oftentimes, many microbes don't grow, and sometimes some microbes grow a lot, so they will take over the culture. And then you send them to sequencing, like 16S sequencing, or maybe transcriptomics or proteomics and so on, okay? But culture is a very fundamental aspect of studies in microbiology. So what we wanted to do is, in this work, we developed a microfluidic device, and this is something we do in the lab all the time, which basically creates a small droplet which can culture an individual microbe, and we can sort these droplets in high throughput. This is still, we're in second 3. Okay, so this is like going in milliseconds. We can get almost 100 droplets per second. And depending on whether a droplet contains a microbial colony or not, we can sort them. As you see, a droplet that contains a colony will be sorted to the top, whereas the empty droplets will be sorted to the bottom. And we can culture these cells in these small water droplets in extremely high throughput. We see here about 20 of them, but we will have 1 million droplets per experiment, and each experiment. So you can see cells grown here. These are empty droplets. These cells have not grown much. They have changed also their morphology. So the advantage of culturing cells in droplets is that we can choose to include only a single cell in each droplet to start with. And when you grow cells from a single cell, this is going to be a monoclonal culture. And then I can take each droplet and then send them to sequencing. Essentially, what we are doing is we're taking individual cells and amplifying them in culture. And this way, we are equilibrating the density fluctuations in a population. Okay, so imagine that if you didn't use the droplets, everything would be cultured in the same dish. They would be competing with each other, and the ones that are growing fast would basically suppress the others, and you would not be seeing these rare species. So with isolated culture in microfluidics, we have seen that we are getting much better representation of the true diversity of microbes in culture. And this also shows in the sequencing experiments. And we were able to, using this enhanced culture environments, detect the existence of antibiotic resistance microbes in many species, in many samples. For example, if you grow these microbiota samples from like donors in plates, you see these microbes, okay? But these are not seen at all. Whereas if you do the same thing in a droplet culture starting from single cells, the diversity is very rich and we start seeing these rare but antibiotic resistant species that are growing in culture. And these are important because these things end up going into patients for microbiota transfusions. And you don't want to essentially transfer or transplant an antibiotic resistance containing sample into a patient, right? Because that could cause issues. Okay, I have one question here, which I just gotten. This is about the sepsis project. Maybe I should answer that before I move on. So, is sepsis currently using technology that does technology can lead to ethical problems? Honestly, I haven't thought about the ethical aspects of this. I'm not an ethicist. I don't see how that's going to lead to ethical problems. We are simply trying to treat patients better based on their individual characteristics, because right now what is being done is you are using essentially the same treatment for everybody. Nobody looks into how the patients differ from each other because it's very fast and people don't have the time to do that. So if we can give doctors the ability of understanding patient-to-patient differences, that can only lead to better treatment. So I do not see how this can lead to any ethical problems. Okay, let's move on. So this microbiome business is important because everybody has a microbiome and it constantly changes and it is related to how healthy we are. So it is important to be able to monitor the microbiota, but oftentimes, especially when it comes to gut microbiota, this involves dealing with stool, which nobody wants to do. So we have recently started a startup company with one of my collaborators, Jack Gilbert, who's a microbiome expert. Essentially, this is an automated tool that collects stool samples over time and does analysis on the spot instead of trying to send these to the laboratory, okay? So this is a startup company trying to commercialize this technology. We have already raised some money for the initial phase of our activities, and it's going well. So there's a lot of commercial interest into technologies that enable these kind of measurements. So maybe I should talk about single-cell virology and then end my slides. So I mentioned single cells. I mentioned that It is important to be able to measure these because population average measurements are not always the most accurate ones. And even before COVID-19, I was collaborating with very talented virologists in my lab who have been interested in different problems in virology. For example, Nir Reiman, who's a trained virologist, he has been interested in HSV-1 infection for a while, and we did some work there. But once COVID-19 hit, we were in a situation where we had a lot of technologies and different analytical techniques which we can use. And this is something we felt we could help. And we started a range of collaborative projects in the past 2 years. One of these things is testing in saliva samples, which we have published already. What I can say about saliva testing is it is better than nasal swabs because the sensitivity is as good, if not better, but it is much less invasive and it can be scaled up. We also did some drug repurposing work, which I'm going to talk about that in the next slides. We did some work on the aerosol transmission of SARS-CoV-2 in hospital settings, especially in the ICU units. Recently, one of the drugs that we discovered to be effective against SARS-CoV-2 infection is now being evaluated in clinical trials, which we are not involved with, but some clinicians in Russia and South Africa has taken it over. And we are continuing to develop other drugs. So, and these are some of the papers that we published in the past year. So let me tell you about this drug repurposing work. So as you can see, this is the cover page from a paper that we published recently in Science that is on drug repurposing for coronaviruses, essentially, including SARS-CoV-2. And what we did in this work is we did essentially a very large screen on existing drugs in their ability to inhibit SARS-CoV-2 infection in cultures. So you might ask, why bother with existing drugs? This is called repurposing. Essentially, you're changing the purpose of an existing drug. And once we have vaccines, why should we bother, especially, right? Well, vaccines are great and they are very effective, but there are these variants that are constantly popping up and they will continue to do so. And it would be great to be able to target the infection both from a vaccine and from a therapeutical side. And when it comes to drugs, so far we don't really have very effective antiviral drugs, unlike we have antibiotics. We have antibiotics, but for viruses, we do not have similarly effective, cheap, and available drugs, okay? So the idea here is to screen existing drugs that have a safety profile already approved. They have gone through clinical trials, the safe doses are known, side effects are known, and so on, right? And if we can find molecules that are effective against the pathogen that we're interested in, then these molecules can go into clinical trials and ultimately to treatment of patients very quickly, unlike a new drug, which needs to go through all the clinical phases, all the regulatory phases, that can take up to several years. So basically, when there's a new pandemic or when there's a new disease, repurposing, if successfully done, can provide a very effective and rapid solution, which new drugs will need to take years and years to test, evaluate, and validate. Question. Maybe I'll talk about this at the end, like all the career stuff and all this question I will talk about at the end. So what I'm trying to say here is new drugs need to be developed. However, we should also look into existing drugs and see if they can be used for an ongoing pandemic. So what we have realized, and many people agree, is that we need effective antivirals that are also safe with known safety profiles, which are also orally available. If you take a pill, you can start using this over the counter at much earlier stages of the disease compared to a drug like remdesivir, which was used a lot in the clinic, which is given by an injection. Okay, people can take this drug only at the later phases of the disease because, by definition, you need to go to the hospital. So that means you're very sick, and it unfortunately was shown that at that stage, antivirals are not making too much of an effect. So we basically need a pill, and that's why we evaluated a bunch of drugs using the single cell techniques that we developed and using also some commercial systems. And we have discovered up to 50 known drugs that inhibit OC43, which is a coronavirus that can be studied in BSL-2. And when we actually measured the top candidates in actual SARS-CoV-2 virus in cultures in a BSL-3 environment, we have found that there are like 20 of these drugs that are really effective in inhibiting the growth of the virus in cell cultures. So one of them is a drug called masitinib, which is developed originally as a tyrosine kinase inhibitor. It is essentially a cancer drug. And people have taken them in clinical trials in the past. There is a known safety profile. People take these for months and months, in the right doses. There are some side effects, but people tolerate it overall fairly well. And the good thing about masitinib is we have shown with crystallography work in collaboration with our partners at Argonne National Laboratories that the drug is inhibiting the main protease of the virus, 3CL-Pro, which is needed for cleavage of the protein complexes and ultimately the growth of the virus. So this is a molecule that is very key as a very important drug target. And this existing drug directly binds to the active pocket of 3CL-Pro and inhibits it. And we also have shown that it is effective in vivo in a mouse model of SARS-CoV-2. It reduces the viral titers in the nose and the lungs by almost 2 and sometimes more orders of magnitude. So it reduces the viral load by about 500 times. Okay, so this drug is very interesting in this regard, and it is a candidate for clinical trials. And we just heard this past week that some clinicians are looking into this. So we are looking forward to getting the results, and hopefully it makes a difference. So future work in drug repurposing or drug development in general, clinical studies are key. You cannot just take a drug because it works in mice or in cell cultures. So these need to be done. We can also design better versions of this molecule, hopefully to reduce side effects of it, reduce the kinase activity, or completely eliminate it. And then we also think that this drug repurposing pipeline should be maintained, developed, and further improved, because this is not going to be the last pandemic. There will be more. And when these things happen, we would like to be ready, at least so that the preclinical work has been done and we can move to clinical trials right away. Okay, so I will not talk too much about the engineering work because I think this is mostly a biology crowd, but I will tell you that we are also developing a lot of devices for analysis of single cells, organoids, culture, live cell imaging, and so on. Okay, here's a microfluidic device that enables you to do extremely high throughput lab on a chip type experiments. Okay, so this is really the kind of interface of electrical, mechanical engineering and biology research. We can use this for understanding neural stem cell differentiation, understanding cellular logic rules, and so on. And we also do a lot of work in basic signaling. Basically here, what we're doing is we are combining experiments with computational work, essentially modeling, to understand how complex signaling systems like NF-kappa B work in infection, autoimmunity, and cancer. So we did a lot of work there and published a bunch of papers in the past. So I don't want to take more time in this regard because it's highly technical set of slides. Maybe I'll just thank my group who did all the work, obviously. This is a very interdisciplinary group. We have physicists like me, engineers, biologists, medical students, chemists, and all. It's a very interesting environment. Everybody has knowledge in different areas, and integrating all this is really exciting. So I will thank my group and all the funding resources, and stop here and take your questions. So maybe I should look into the chat and start answering from the last question. So the question is, biomedical engineering is important has been in the past pandemic. Do you have any suggestions for people who want to go into bioengineering, do bioengineering research? Sure, I have suggestions for such people. I think it's a very interesting and important area. It's going to continue to be interesting and important, even more so. As you know, the 19th century, you could argue that it's been the century of physics, the 20th century, maybe the chemistry. The 21st century is probably the century of medicine, biological research. Computing, of course, is very important too. So I would definitely recommend this as a career track for everybody. There are certain things that one needs to be careful about. One is, as far as I know, bioengineering research in Turkey is very limited. So you should be looking into going abroad, okay? Europe, Asia, USA, wherever. But there's a lot of opportunities for people who want to go into bioengineering, but most often in other countries. So you should be willing to do that. That's number one. For that to happen, you need to, of course, in Turkey, you need to be able to write, read, and speak in English. So that's something you can start working on right away. And lastly, I understand most of the students here right now are coming from the biology background, and I'm sure you're getting a very good biology training. What you should focus on, I believe, is also getting skills and training in computation. Because if your skills in computation are lacking, it's very difficult to do biology or bioengineering research these days. So, and these things take time, so I would start working on them right away, build your skills, be good at programming, data analysis, data science. You don't want to be somebody who has only wet lab skills. You want to have some computational skills. So basically what I'm saying is if you're getting a good basic biology training in Turkey, you can speak and write and read in English, and you can also program. The world is your oyster. You can get a variety of jobs, both in academic and industrial settings all over the world, okay? These are my suggestions for people who want to do bioengineering research and take a job in that direction. So what led me into biological research? So like I mentioned, I have always been in physics and engineering. And when I finished my PhD, I wanted to continue doing research. And physics is very mature. The fundamental developments have happened. We have good theories. We have a very good understanding of the physical world. The things to be done, the new things to be done in physics are really at the cutting edge, yes, but there are also like small increments of compared to what has happened in the 19th century. So let's put it this way: we will not have a second Isaac Newton or Albert Einstein, right? Those kind of fundamental big things will not happen again. So when you look at biology, it's wide open. The theoretical approach is just becoming popular, right? And the understanding, although there's a lot of information, the basic understanding is still lacking, and there are a lot of opportunities to do really high-end, cutting-edge science in biology. So that's one of the things that really attracted me to it. And the second thing is I have seen biology research really benefiting a lot from computing, engineering, and so on, which I could do myself. And I saw this opportunity in that direction. And lastly, biological questions are really fun. You know, they are not unlike physical problems. When you're dealing with cell signaling, I can get any engineering student become excited about cell signaling, even if they haven't done nothing about biology before. So there are a lot of overlaps, and I have seen that, and I became excited. And of course, the way it happened is you have to work with a good mentor who's done the transition themselves before. I was able to do that at Stanford, and yeah, that's how it happened. Okay, do you suggest double major or any minor with molecular biology? Which areas do you suggest? I don't suggest major or minor. What I'm suggesting is get a good base education in a particular field. You're a biologist, great, learn that. You're an engineer, great. You're a physicist, great, whatever. But then learn how to do computing, okay? And definitely improve yourselves in your abilities in English, and then apply for graduate schools. And don't feel inhibited in any other way. You don't need to have an engineering minor to apply into a bioengineering department. Nobody cares. What we will care when we're selecting students is, do you have a good base education in a particular area? And then do you have the important skills of language and computing? If these are there, you can apply to any department, any institute, any field, and start doing research. Which school in my career influenced you the most, me the most? My postdoc enabled me to switch to biological research, so I would say my time at Stanford Bioengineering was the most influential. What should be the process plan? Okay, so this is about setting up a business. What is the process to set up a business? What are the steps? Do I have any tips for a young entrepreneur to start their own business? It all depends on the environment that you're in. If you're in Turkey, I don't know what it takes to start a business in Turkey. Probably you need to know somebody higher up, have connections. That's the most important thing. If you're in the US, connections also help, but the most important thing is you need to have a good idea, and then you need to start putting time into it, develop that idea. That's number one. Number two, you need to convince funders to give you money, okay? Almost all businesses start with some level of investment from an investment company or government or some philanthropist, right? So you need to be able to sell your idea and then bring in the initial seed money to start your business. Now you need to have good partners. In my case, I'm at the university, I am a professor. I will not be able to put 100% effort into this, so I need people who can do that, right? I need probably a CEO who's willing to do this coming from a business end. So having the right partners is important. Having the funding is important, and having a good idea, of course, is the beginning of everything, but it's definitely not everything itself. a good idea is the start, but you need so many other things to come up with all that. And you also need to have luck. And that means even if you have all the right pieces, your business may not succeed. And if it fails, you should have a backup plan, maybe your education or whatever. And you need to be risk tolerant. If you cannot tolerate that risk, maybe it's better to just stick to the traditional route, go start with a company, work in somebody else's business. But if you're really risk tolerant, you're willing to risk everything and put 100% effort into it, then go ahead and do it. I'd say these are the important aspects. How did the process of co-founding BiomeSense happen? So it's very simple. I came to Chicago. I met Jack Gilbert, who's my partner in this. And Jack had the idea. He found a CEO. We started a company, the three of us. The starting of the company is easy, but then you need to start raising money and doing some work. That's the difficult part, right? There's a lot of logistics. There's a lot of procedures. You also need to deal with everything in the university environment. But again, it usually starts from having the right idea and finding the right people. And once you have that, everything else can go much faster. Okay, what is there a difference in knowledge and perspective that you feel in the assignment? Okay, so yeah, there are differences in perspectives and skill sets and approaches of people coming from Asia, from Europe, from the United States, and from Turkey. I'm not going to go and do a comparison. What I can say is, coming from Turkey, you will have a good base education, that's for sure. If you're in biology, you'll know biology. If you're in physics, you will know physics. What you probably need to do is improve yourself in two areas that your formal education may not give you. For whatever reason, in Turkey, we are not able to educate people in language skills. I don't know why this happens. I have some theories, not need to get into that, but you need to make an effort to be able to write, read, and speak in English, because like it or not, this is the main means of communication in the world right now. This probably is the most hindrance, and this requires effort from an early age, so that by the time you graduate, you feel confident about that, right? So Turkish students need to spend some time on this and think about this. And the second thing is, sometimes in our traditional programs, say if you're biology, you may have very little exposure to other disciplines, maybe in computing and engineering. You need to make an effort to have at least some opinion about these and maybe have some basic skills. So these are the two things that you need to make an extra effort if you're from Turkey to be ready for taking over any kind of job in the world. And if you have these, feel confident, you'll be fine. In basic knowledge, you will not be missing, but in these two areas, there might be some deficiencies. I think this is it. Thank you very much. I mean, if somebody wants to ask a question, just go ahead. Yeah.
Seems like we don't have any more questions.
Okay.
Thank you for joining us.
Thank you. Thanks for the invitation.
Thanks for the great presentation.
Thanks. Okay, everybody have a good night.